Recording. Excellent. So welcome to this first WNT webinar on essentials of validation principles and good in vitro method practice. So we have about an hour and 15 minutes for this webinar today. Here is the program. So we will have Valerie, Zuang and Ingrid Langezal as our first speakers. Then Laura Rossi from ECHA. Then Anna Lowit from the US EPA. Then Deborah Ratzlaff from Health Canada. And our last speaker will be Elijah Petersen from NIST in the US. So as explained, this is a foundational webinar setting the scene, the status quo, reminding ourselves of the essential elements to validate standardized methods. And it will be the occasion to hear perspectives of regulators also who are speaking today on what they value most in the validation process and principles as a quality assurance system that guarantees that a method will produce and reproduce trustable data. And what do they need to know about a new method for a new endpoint? So as I said, the webinar will be recorded. And
sorry, I'm not sure if you're sharing your screen or if you're sharing something, but I can't see it, and maybe others can't as well.
Yeah. Okay, you should be able to see the second slide.
Yes, good. Yes,
thank you. Thank you, Natalie, for reminding me. So the webinar is recorded. It will be available later in early July on the WNT community site, like other webinars in the series. Attendees may use the chat box for discussions, raising questions, and the WNT will have an opportunity for some follow-up in writing on presentations, questions, and comments raised. There will be a short webinar report prepared by the Secretariat and the steering group as material for the WNT workshop that will be held in December this year. So without further ado, I'll now hand over to our first speaker, Valerie. I'll switch to your presentation, Valerie, in a second. Please go ahead, Valerie.
Okay, thank you very much, Anne. So good day, everyone. My presentation will be about some essential principles of validation, some lessons learned during validation studies, and some challenges and how they could be addressed. My colleague Ingrid Langezal will then follow up with a few key messages related to good in vitro method practices. Next slide. Next slide, Anne. OECD Guidance Document 34 says that the rationale for test methods should be available, the relationship between the test method's endpoint and the biological phenomenon of interest should be described, a detailed protocol for the test method should be available, the intra- and interlaboratory reproducibility of the test method should be demonstrated, the test method's performance should be demonstrated and based on the testing of reference chemicals representative of the types of substances for which the test method will be used. The performance of the test method should have been evaluated in relation to relevant information from the species of concern and existing relevant toxicity testing data. Ideally, all data supporting the validity of a test method should have been obtained in accordance with the principles of GLP, and all data supporting the assessment of the validity of the test method should be available for expert review. The fulfillment of these criteria would then facilitate acceptance and standardization of the test methods. And worth to mention is also that these criteria apply to both in vitro and in vivo test methods. Next slide. In the same guidance document, validation is defined as the process by which the reliability and relevance of a particular approach, method, process, or assessment is established for a defined purpose. A test method or approach is never valid in an absolute sense, but only in relation to a defined purpose. Next slide, please. Validation can thus be seen as the ultimate means to build confidence and reach acceptance, but also to ensure that solid science-based decisions are made to protect human health and the environment. Validation facilitates the development of globally harmonized test guidelines based on standard methods. And as such, it should not be perceived as an impediment or constraint. Acceptance cannot simply be imposed, but needs a process of trust building amongst various actors. Next slide. Thus, the principles of validation, that's to say that a method or approach should be relevant and reliable, are universal and valid. The validation process is adaptable and depends on the purpose. One size does not fit all. The process needs to be fit for purpose. Next slide. Nowadays, we are faced with a plethora of new technologies such as cheminformatics and computational models, including machine learning and artificial intelligence, organ-on-a-chip devices, high-content imaging, omics technology, 3D tissues, exposure modeling, and so on. All the data generated is then integrated to predict safety. Furthermore, we are experiencing a convergence of different scientific disciplines such as biology, engineering, physics, electronics, and so on. So validating or standardizing an approach starts to have a broader meaning depending on what it refers to. Next slide. Several workshops on validation had been organized in 2017 and 2018. In 2017, the Dutch RIVM and the German BFR organized a joint workshop where the drivers and barriers concerning implementation of innovative 3Rs approaches in regulatory toxicology were discussed. This was followed up by a second workshop in 2018, aiming to develop feasible solutions and recommendations to move innovation forward in this area. In 2018, we organized a workshop with our ICATEM partners. ICATEM stands for International Cooperation on Alternative Test Methods. And we invited some additional governmental institutes and authorities, including the OECD. At the ICATEM workshop, we discussed the lessons learned by ICATEM partners in the conduct of validation studies. Click, please. Then we discussed 4 challenging topics, in particular, how to establish relevance without animal data, how to evaluate reliability without ring trials, how to evolve validation practices, and how to develop the concept of standards. Next slide. For the lessons learned, here are some scientific issues we came across. How should the relevance of single methods be assessed if you know that a method needs to be integrated with several others? Relevance assessment should be made within that integration context, and one should not emphasize on the relevance of the individual methods. Single methods validation is also time-consuming and costly, in particular if methods are not meant to be used individually. Another issue is the poor or unknown quality of the reference data, which are often in vivo animal data, against which you validate your in vitro data. And related to this, a too high focus is given to the animal test as the gold standard and the derived data, which are used to assess the predictive capacity of your test, when animal data are not always human relevant. Another issue is poor chemical selection, introducing bias. For example, when the same chemicals are used for the development and validation of the test method, sometimes the same chemicals are even used for transferability and training purposes. Further guidance on chemical selection is needed, how this should be properly done. Then lab-specific SOPs that hinder transferability. For example, the SOPs are too specific to one lab and too detailed with regard to the material that should be used. Next slide. Regarding non-scientific issues, the current regulatory requirements are mostly based on animal studies, whereas the new non-animal methods provide a different type of information that does not fit these requirements. International harmonization is critical, but having guidelines does not guarantee use by regulators and industry if they do not fit the regulatory requirement in a jurisdiction. Engagement with stakeholders at every step is important. A review and evaluation of uses and needs of data across end users is key to define fit for purpose and context of use. Tackling intellectual property, ensuring commercial availability, and avoiding abuse of monopolies. This is a topic that was discussed considerably over the recent years at the OECD and addressed with the development of the guiding principles on good practices for the availability, distribution of protected elements in OECD test guidelines and the implementation of fair, reasonable, and non-discriminatory different licensing terms and conditions. Lack of transparency of protected elements, for example, when a test method includes confidential business information and the prediction model is not explicitly described. GLP implementation of protected elements and cloud-based computerized systems, also a topic that has been discussed recently in the OECD GLP Working Party. Next slide. Regarding the challenges, one which came up on several occasions was how to establish relevance without animal data. While the use of human data is recommended whenever available, a good mechanistic understanding of the endpoint, for example, via an AOP, is key. This is more difficult for complex endpoints where all relevant mechanisms might not be known, or when some modes of actions are triggered by only a few chemicals. In that case, when can we trust a negative result? Provide justifications for not comparing to animal data by characterizing the animal data and understanding its limitations, like variability and limitations to predict human adverse effects in the case of human health endpoints, of course. Next slide. Another challenge is how to evaluate reliability without necessarily always carrying out multi-laboratory ring trials, which are costly and time-consuming, as we have seen before. Demonstrating reproducibility remains, however, essential. There could be a better characterization of within-lab reproducibility and of the critical steps by the test method developer, for example, by a sensitivity analysis of all the parameters that can affect the results. Training and transferability are critical steps because they provide an opportunity to discuss and improve clarity and independent implementation of the protocol. And if they are well performed, reproducibility assessment could be done with a small number of coded chemicals and perhaps in only one naive laboratory. Proficiency testing adds confidence on the capacity of a lab to perform a test. and following good in vitro method practices early in the process, for example, during the development phase of a method, already ensures a high reproducibility of a test method. Next slide. Regarding validation practices, further guidance for external validation studies may be needed, as to avoid that test method submitters come with a complete package full of shortcomings, and there is no more funding left to acquire additional data and evidence. It is not always clear from the beginning how to combine a mechanistic method with other methods in a defined approach or an IATA context for regulatory applications. In that case, it could be considered to decouple technical validation and biological relevance from regulatory application, for which case studies could be developed first. The development of common reference chemical sets to facilitate defined approach development and evaluation is very important. Narrowing the purpose of a validation study, for instance, by focusing on specific regulatory requirements and defined chemical spaces without trying to cover the universe of chemicals in one study. Whenever possible, relevance should be assessed ahead of ring trials. Early dialogue with regulators and industry to prioritize methods is recommended. Yeah. And the creation of standards to facilitate assessment of new or fast evolving technologies. Next slide. In that sense, would it be possible in future to define standards for classes of methods rather than standards for very similar identical methods as it is currently done? This could be evolved performance standards where essential test method components could be key features that need to be characterized rather than a prescriptive description of how a method should look like. Reference chemicals could be a description of the chemical space to be covered, and better ways to assess performance instead of target values could be explored. In the mid to long term, could the OECD test guideline program evolve towards the development and acceptance of standards so that these, rather than methods, are covered by mutual acceptance of data? Next slide. Some final thoughts. Validation will remain one of the cornerstones of a successful toxicological evolution. However, validation needs to keep pace with rapid scientific progress. It is important to keep scientific integrity, credibility, and usefulness while making the process more efficient. Validation should be framed as a process to characterize and reduce uncertainty rather than a ring trial to demonstrate toxicological equivalence to the animal test. And after 17 years, is guidance document 34 maybe candidate for being updated? Thank you. I hand over to Ingrid now.
Okay, thank you, Valerie. Yeah, so during this webinar, I would like to share with you some key messages for the application of good in vitro method practice that we are always sharing with consortia that are asking us to be trained in good in vitro method practice, and we are also applying, of course, GIVIMP during our own validation studies with the current validation study for the thyroid disruptor methods. So the guidance document GIVIMP, Good in Vitro Method Practices, is available since 2019, and it has the main scope to provide guidance for the development, use, and implementation of in vitro methods. It's a tool that really helps to implement good practices already early during the method development process when they are destined for regulatory use. Click on. So when it is properly implemented, we think it will increase the credibility of mechanistic data, increase the reliability and the integrity of the generated data, and will improve the efficiency of method development and use for regulatory purposes. Next slide, please. So one of the key messages that we always try to transmit is that the method developer should demonstrate that the method is specific for the mechanism to be assessed. Next slide. So one very important element of a method is, of course, the test system. And for cell-based test systems, it's important to measure the specificity. So this is an example from the thyroid validation study where the original method was using NTHY ori cells, which were human thyroid cells, for the measurement of TPO inhibition. Click, please. So in the publication, there was a graph presented, and there was the conclusion that these thyroid cells were actually containing thyroid peroxidase, whereas, for example, liver cells, non-thyroid cells, were not having any peroxidase. Yes, click. So in our thyroid validation study, we have actually changed the test system to another cell line that also had a control cell line. And this was the thyroid follicular FTC238 cell line that was transfected with human recombinant TPO. The test system supplier confirmed, as you can see here on the plot, that the wild type cells did not contain any TPO and the transfected cells did. But when then the EU NETVA laboratory was actually testing a chemical for TPO inhibition with both these cells, there was not really a difference seen between the wild type cells and the transfected cells. And therefore, it was finally concluded that the method could not really discriminate thyroid peroxidase from overall peroxidases. If you click one more time, you can see that according to the scientists, the luminol substrate was not able to discriminate TPO from other peroxidases. So basically, we stopped then the assessment of the method. Another key message that we always give is that it is important to really control all the essential elements of the method. So that is, it goes from basically all the elements, so the materials and reagents, etc. Next slide. So one thing we always emphasize is that GIVIMP is discouraging the use of serum because serum can it contains a lot of unidentified components, and it is proven that also the variability of the results is increasing. So we are always asking if in the case that there is a chemically defined medium available, to use that, because then you don't need to use the serum. But the challenge is to do this in practice, because in fact in our battery of thyroid methods, there is not one single method where the serum could be really replaced. So this is still a challenge for the future. If you click one more time, then you can see that we also discourage the use of antibiotics, because they can mask bacterial infections and can also disrupt cell biology. Next slide. The most important component of an in vitro method is the test system. And when it is a cell-based test system, we always emphasize that if you click a few times, that the information should be obtained from the supplier on the identity and the purity of the cells. And in the case that the supplier is not providing this information, which is nowadays it's very common that the supplier provides everything, but it could be that the cell line is obtained from a collaborator, and then we really recommend to confirm identity of the cell of human cell lines with STR profiling, and of animal cells with DNA barcoding. Then below you see some examples. What can be the consequences of misidentified cell lines? And one you know of yourself, because in an OECD test guideline, actually in a very last stage, in a very late stage, it was found out that actually the cell line that was always reported was not the true identity of the cell line. So the guideline had to be updated. So this, we always emphasize it's important to do. Next slide. Another key message is that we always, GIVIMP is also saying that you need to confirm that the results are reproducible within your laboratory. Next slide. So this is an example of an experiment, of several experiments that we have done ourselves in-house with the ARCALUX method. And we use this then as an example to show a reproducibility experiment. And we always say it's important to consider different batches of the test system and of some critical material and reagents, and that the experiments should be biologically independent. Next, click. And that it is very important to, as soon as possible, when you have historical data, set acceptance criteria, because then you can really also monitor if any changes to the method have an impact on the results. So acceptance criteria should be set for the test system, for the reference item, which is usually a positive chemical, for control items, and also once the method is fully developed, acceptance criteria should be set for the validity of test item results. Next slide. So the final key message is that we always say is that when you develop a method, you need to also keep in mind any future transfer of your method to another laboratory. Next slide. So for that, what we have been doing quite consistently is really check that all the elements of the method are also available to others, and we follow there the OECD guidance documents on the IP elements, and we map really the IP elements that are covered by a method, and estimate if there's anything that may block future availability. And we also what we did for our thyroid validation study is also have a written commitment from the suppliers that they are willing to respect the friend conditions and make everything available at fair, reasonable, and non-discriminatory conditions. Next slide. So now I give a few things that are basically mentioned in the different sections of the GIVIMP guidance. That it is important to write detailed SOP that are fully describing the methods, and that the elements should not be really facility specific, so that they can also be used by other laboratories. It's important to define critical steps in the procedure, and that you need to have historical data and process data available. And it is important to store your records for future use. Click. And we also give recommendations for references that can be used to know what to report. So, for example, OECD harmonized templates when there is already test guidelines. And also we always recommend people to look at SciRep tool from the Karolinska Institute, which is really a tool for researchers, but also for reviewers to see how complete a method is reported with respect to elements that are needed for regulatory use. So with that, I would like to conclude by giving the reference to the GIVIMP guidance document in the case you would like to read more. And I hand over the word back to you, Anne.
Thank you very much, Ingrid and Valerie. We'll move next to Laura. So Laura, please go ahead and don't hesitate to add your comments or questions or any thoughts you may have in the chat box. Don't be shy. Go ahead.
Thank you, and good day from my side as well. My presentation is briefly touching upon how new approach methodologies can be used under REACH and what are the issues that we currently see. Next slide, please. So how the REACH regulation has been built is that under REACH, one needs to provide information on the intrinsic properties of the substance. And that data, it has to firstly demonstrate the safe use of the substance, but it needs to be adequate also for classification and labeling of the substance according to CLB. And how the legal text has been drafted, this poses currently some restrictions how NAM-derived data can be used. For us, we see that the OECD adopted method currently can be divided into 3 tiers, which are, for example, the standalone methods, like skin corrosion irritation in vitro studies. Of course, depending on the outcome, you might need to do both of the assays, but in some cases, one assay is enough. Then we have the partial replacement methods. As an example, the skin sensation adverse outcome pathway-based methods. However, depending on what methods have been actually performed, currently a defined approach can be applied, and depending on the outcome of that, that can be the full replacement of the respective in vivo method. Then as a third tier, we see the screening and prioritization methods, which are, for example, the in vitro methods for endocrine disrupting chemicals, such as the TG455. Next slide, please. What is also embedded into the REACH regulation is the 3 Rs. And how we are implementing it, these are some examples, is firstly, the effective implementation of our integrated regulatory strategy, which is firstly the forced data sharing at registration. And this means that for one substance, only one set of data should be generated if new data needs to be generated. Use of new approach methodologies for priority setting, addressing chemicals in groups, and this looking chemicals in groups will help us detecting those chemicals where we would need to ask for additional testing or to see whether we have that type of chemical universe covered. And then, of course, development of testing strategies based on read-across. Then the second pillar is investment in international activities that promote alternatives. So we are heavily involved in the development of the OECD QSAR toolbox. We support different research flagship projects, such as EU TOC Risk and PARC, and we do actively contribute to the ECHA project. And as the third pillar is making data available. So all the data that we have in our registration database is made available via our dissemination site. And this is important also when developing new alternatives for data generation. Next slide, please. So what is important when looking at NAMs and regulatory acceptance for us? And many of these have been raised already by Valerie. So we have the reproducibility and standardization of the tools and harmonization, how the data is handled and how the NAM data is reported. The method has to be reliable. We need to know how well does it perform and what are the possible limitations of that new method. Relevance. How can we use that data in regulatory decision making? So would it be a standalone method, partial replacement, or a screening method? And how does that NAM derived information is in comparison to our current regulatory methods, which are the guideline studies? Is the method, does it cover industrial chemicals? And does it have a defined applicability domain? Then, of course, the confidence, it comes from the scientific acceptance and validation that is performed by different validation bodies, but also the legal certainty that we have in the use of NAM data. And I will come to that a little bit later again. So can you use that data to cover the information requirement? Next slide, please. So here I will focus a little bit more on complex systemic endpoints. And at this point of time, we see quite some barriers in considering NAM data as the primary input for definitive hazard assessment under REACH and CLP. And here I have listed a couple of the difficulties that we have. The REACH requirements currently refer to animal tests. And for some of the information requirements, a specific OECD in vivo test guideline is even indicated in the annex. The REACH allows to use adaptation. So instead of providing that in vivo study, you can try to provide additional information, and normally that is weight of evidence or read across. But and in this case, in future, perhaps also NAM data. But if adaptations are used, it is assumed that equivalent level of information needs to be provided as with the standard information requirement, and it has to be also suitable for risk assessment and classification labeling. With the current in vivo systemic toxicity studies, you measure spectrum of different type of endpoints. You have the clinical observations, hematological data, clinical biochemistry, pathology, and histopathology. And at this point of time, at least the NAMs cannot replicate or provide equivalent level of information for these wide effects. We also need the NOAEL and LOAEL values based on observed adverse effects. And there is currently a limited number of NAMs that are able to predict those adverse outcomes. And if those that are available, they can only cover a limited number of those effects. And this is also what was raised in the previous presentation. So if you have a different type of NAMs, when do you know that you have enough data sort of to make a decision what is the hazardous profile of your substance? We know that the NAMs are very useful to confirm or support hypotheses about mode of action in general. However, for industrial chemicals, there is often not so much knowledge or data available. So in REACH registration database, we rarely see broad NAM data. So that is not provided to us. And at this point of time, and one reason of this webinar series is as well that currently majority of the NAMs have not gone under formal validation and are not approved in the OECD test guideline program. And therefore, at the moment, they don't satisfy the principle of MAD. Next slide, please. And here I just wanted to provide an example what comparable with, for example, with repeated dose toxicity means. So in case one wants to adapt from the standard information requirement, one needs to provide the comparable information. So, and here the NAM data would need to demonstrate that the outcome is comparable with the repeated dose-tox studies in context of hazard characterization and risk management. And therefore, the NAM would need to provide estimated NOAEL and LOELs. So the NOAEL is the potential source for systemic DNL derivation, and the LOEL value is to be used for stock repeated exposure classification. In addition to that, the standard 90-day study provides us triggers or indications of reproductive toxicity, immunotoxicity, neurotoxicity, carcinogenicity, ED-related effects, and developmental toxicity. So there is a multitude of information what we do gain from currently from the animal studies. Next slide, please. So for us, what are the important aspects concerning validation outcome? So for us, it is important that we know the scope or the end use of the new approach methodology or the approach. It needs to be clear. So is it intended to be used as a full or partial replacement method or as a screening method to aid in testing prioritization, or in the future combination with other methods, for example, as a defined approach? Can that method or approach be used to determine a particular intrinsic property of the substance? And what is also important is that the method needs to be robust, reproducible, and to have information on potential limitations, for example, the applicability domain. And this, of course, is looked in the validation, and as explained also in the previous presentation. What we also then at the end of the process think is important that the method or approach is included into the OECD test guideline program and then hence covered by MAD, because this would ensure harmonization in data generation and able the use across different regulations. So all of these points that I raised above are important, but of course I'm not excluding others in generating legal certainty. And this is something that is very important for us in ECHA. So if we are to request NAM-based testing from industry, or industry is generating NAM-based data, there needs to be a certainty that such data is fit for the purpose and will generate the data needed. So for example, we can't go and ask a company to do 5 different studies, and then when they have provided the data, we are in the situation that, sorry, this didn't provide us the information that we needed, and you still need to do the original test. So there has to be legal certainty for all parties that the approach that is being used or requested is suitable for the purpose. I think that was my, yes. So thank you. I tried to be short and raise the main aspects that we have in the use of NAM-related data. Thank you.
Thank you so much, Laura. And we will now move to our next series of speakers from North America, and starting with Anna Lowit from the US EPA. Please, Anna, go ahead. The floor is yours.
Hello, everyone. Thanks, Anne, for inviting me to talk. So I'm going to go pretty fast. I'm going to cover a lot in a short ground, but I think you'll hear a lot of themes similar to the kinds of things that Valerie was talking about earlier. I think there's a pretty strong alignment between our ECFAM partners at JRC and some of the things going on within the US, both at ECFAM with an I, but also across some of our agencies. Oh, can you go back? There's a bug flying around, by the way, if you see me swatting. So just one point on the slide, if many of you didn't know this, that many of you know that for many years I've been in the pesticide office at EPA. I've been on a temporary a lateral job change for the last 6 months or so to our toxics office working on industrial chemicals. So I have been doing some different things in the last few months. Okay, next slide. I am using the disclaimer this morning, and normally I don't do that because usually when I speak, I'm talking on behalf of EPA on official government policy, but you'll see most of the things in the slide deck are actually work that's in process or things that are in draft form. So I have to use the disclaimer. But just because the disclaimer is there doesn't mean that it's not the prevailing view of most of the scientists at the Office of Chemical Safety and Pollution Prevention, and also many parts of the Office of Research and Development. Next slide. So just a couple of thoughts on the current process. I think Valerie did a really nice job of covering some of the details and a lot of the history. So thanks to Valerie. So I didn't have to do a lot of that. So I'm going to just sort of jump over that. So, you know, our idea is that the fundamental principles outlined in GD34 really still hold true. You know, the concept of relevance and reliability are really foundational to how we think about validating methods and building confidence in them for regulatory use. But we all acknowledge that there's a pretty substantial delay between the development of a method and its regulatory uptake, not only in our regulatory agencies and our process, but also in industry end users with respect to building confidence. And we certainly think that some of that has to do with the early steps in the process and the need to define a new approach method's purpose and its fit for purpose, as we'll call it, much earlier in the process, and to engage end users, whether they're government regulatory people, either at the federal level or here in the US, also at the state level, but also our industry end users who have to use them in-house for research and development of their chemicals. Excuse me. That, you know, I think there's a strong acknowledgement that interlaboratory ring trials, although useful and has been our historical approach, are really not building the confidence that we need in the methods. And in fact, we're, as many of us have acknowledged, that where there are OECD guidelines for which we're almost revalidating in many regions. Really good example is in the pesticide space, the Office of Pesticide Programs, and to a lesser extent at EFSA, because there are not very many pesticide active ingredients and no formulations in the studies and in the ring trials. A lot of that work is almost being redone from scratch, using chemicals in those spaces, either in the active ingredients or in the formulation space, to build more confidence in their use. And we're finding actually that some of those methods aren't working that well. And so we have guidelines that aren't actually doing the things that we need from the regulatory space. So we really do need a new way about going about this. And the other thing that we want to comment on that I'm not sure Valerie hit is that although GD34 allows for a flexible and modular approach, it's not actually the way that it's carried out. That the interpretation of those studies has become very rigid and is really looked at as a checkbox approach as opposed to something flexible and modular. Next slide. There's a group of us, several of whom are on this call, including Anna van der Zalm, who's the first author. I noticed Warren Casey is on, and Nicole Kleinshore. And I haven't seen patients as well yet, but there's several of us on the call have been working together on a proposed framework for thinking about building scientific confidence and validation in new approach methods for regulatory use, and have a manuscript that we just submitted about a month ago, actually. So we're looking forward to getting the feedback back. But I thought I would take just a couple minutes and talk about the concepts in that paper. Next slide. So I'm going to show you a couple of figures that have been extracted from that that were initially developed by Warren Casey as part of our paper. So the framework has several characteristics that are really the foundation of validation. And none of these should look different or unusual to anyone. They're actually the same foundational pieces NGD34 and the Casati ICADM paper from the workshop that Valerie talked about a little while ago. They're the same foundational pieces in the annex to the new DAS guideline for skin sensitization. But what we want to do in this framework is to think about reordering them and repurposing them and maybe moving the emphasis around a bit. Taking the emphasis away from one-to-one comparisons to animal data, where there's all sorts of challenges that come with that and often send us in the wrong direction, and move the emphasis more to early steps in the process where we invite and encourage method developers to work directly with end users. So what the framework is really built on is starting with this idea of fit for purpose. Other people, you can also use the context of use concept. It's basically the same thing, that you start with a very defined use, use and purpose for your method and how it fits into the regulatory framework and under what conditions. So there's a specificity there, and there's a connectivity there between the method and the questions that are being answered. That's in reverse to most of what happens now, which is the method developer has a particularly good idea, they get some funding to do a method, and then they hope and cross their fingers that someone will pick it up and it'll be useful for something. And that's really opposite to what should be happening. Our framework puts a lot of emphasis on the use of human biological relevance and the use of adverse outcome pathways and understanding human biology as more important than that direct comparison to the laboratory animal data, which is wrought with challenges, right? Well, because we have the animal to human extrapolation. There's increasing literature that animal studies often do not predict human outcomes. There are, challenges of lack of genetic diversity in those animals, et cetera, et cetera. So we're putting more emphasis on understanding human biology and predicting human biology than we are in the animal studies. Next slide. So a little bit more sort of focus on this fitness for purpose idea or context of use, depending on your regulation and your agency. So for example, so if you walk through the ideas, it would be really starting at the ground up. Specifically, what regulatory statutes would a particular method be applicable to? So here in the US, you could be talking about TSCA, you could be talking about any of the pesticide regulations. There are other regulations relevant to the food, to FDA, Consumer Product Safety Commission, or even European legislations, for example. Shutting the chat down, it's distracting. Then you would think about is in the particular new approach method or the information, how is it, or to what extent is that information relevant or useful or sufficient to answer our regulatory endpoints? So then we get into an explicit explanation of the strengths and the weaknesses of a particular method, but you also have to compare those strengths and uncertainties to the animal method, because let's say a new approach method, may not cover all of biology, but if the method we're using for the animals is actually not human relevant in its method or its outcomes, which is better, a human endpoint that it may not be complete, or something that's not human relevant at all. And that's one of the things we don't do right now, is that explicit comparison of the strengths and uncertainties of those different choices. Then there's an explicit explanation of how you would use a method. Can it be used alone? Does it have to be used in combination? Can you combine it with computational ideas? Is there an IATA, for example? Where in the risk assessment or risk evaluation process does this lie? We're talking about screening, prioritization, hazard identification, characterization, grouping, et cetera. And this explicit and thoughtful approach to understanding the method at the beginning will far more ensure that our methods are useful to people like myself and many of you in our regulatory programs. And we believe that by doing that, we're going to have application and acceptance much faster. Next slide. So a couple of related things that help us with the thought process of this. A couple of years ago, under the direction of Warren Casey, the U.S. Interagency Coordinating Committee for the Validation of Alternative Methods, otherwise known as ICVAM with an I, developed its strategic roadmap a few years ago. And if you've never read it, it's actually a really nice read. It's a really short document, really conceptual in its thought process, but the simplicity of it is actually its power. That the roadmap is really built on this idea of moving away from the linear, stepwise approach of the ring trials with this assumption that we would be moving towards regulatory acceptance. And that linearity process just doesn't work. That what we really have to build is an iterative, collaborative relationship and process that starts with, that really integrates the developers and the users. So the three pieces there are technology, utilization, and confidence, and they're really interconnected. So if you look at the piece in the red circle, this idea of bringing method developers directly in contact and collaboration with the end users at both the federal, the state regulatory agencies, but also industry end users is really the key to getting the whole process started. And those ideas really come from this, in my mind, the strategic roadmap. Next slide. So just to close, a few things going on here in the US related to these issues. We're very active here in the US in this idea of working towards a more scientific, hypothesis-based scientific process for building confidence, moving away from the ring trial process that's not answering our questions anyway. So there's some big things that are happening that you may or may not be aware of. The National Academy of Sciences has been contracted by the EPA to develop a new report on a variety of factors. One of those pieces is to do a review of validation and scientific confidence frameworks. There have been two relatively recent workshops. There was one in late 2021 and one again this spring. And a couple of people on this call are actually members of that NIST committee. So we'll be looking forward to that report in the next year or so. At ICVAM, we do have a validation work group that is in the middle of rewriting the ICVAM validation technical guidance. And I think people will find that the concepts in that guidance, in that document, I don't think we're going to call it that guidance because then we've got O&B problems, that document will have many of the exact same concepts and the Van der Sal framework paper that I talked about a few minutes ago, and that's very purposeful. Several members of ICVAM are actually co-writers on that paper, including myself, and John Gordon from CPSC, who is one of our new chairs, by the way. Just specific to EPA, we have a couple of things that we're doing as part of our new approach method work. Many of you will remember in 2019, our former administrator, Andrew Wheeler, had put out a directive to the Office of Research and Development and the Office of Chemical Safety and Pollution Prevention to work together to advance new approach methods at EPA. And we've been working hard on that directive since that time. And there are two activities that are ongoing right now related to that. We updated our work plan in late 2021. And one of the milestones in that work plan is actually the NIST review that's being conducted. One of the other milestones in that is the development of a validation framework for use by EPA. And I think you'll also find that's being worked on right now. Allison Harrell is the primary author, but we have a work group of people working on that. Several of the people on that work group are also co-authors on the Van der Zalm paper. So there's a very intentional use of the same concepts across the EPA validation, confidence building framework, and the ICFAM one. So that there will, the idea being that we'll have a pretty strong US support of using these concepts of fit for purpose on top of all the other ones. The other thing to point out, if you're not aware, EPA is hosting our third conference on the state of the science for use of new approach methods in the middle of October. That got announced in the last couple of weeks. So if you just Google that, you can find the announcement in the draft agenda. We do have an entire session of that meeting dedicated to validation and confidence building. There'll be a little bit on the status of the NES report, but we also have some speakers talking about modern approaches to validation. So I'll stop there. I think that's my last slide.
Thank you so much, Anna. Without further ado, I'll move to our next speaker from Health Canada, Deborah. Putting your slides on. Please go ahead, the floor is yours.
Thanks, Dan. Hi, I'm Deborah Ratzlaff. I'll be talking today about Health Canada's new substances program and the validation of NAMs. Next slide, please. To give you some background information, in Canada, a new substance is a substance that's not listed on our domestic substances list. We work under the new substances notification regulations, and the program is responsible for administering the NSNR regulations under CEPA, the Canadian Environmental Protection Act. These regulations ensure that new substances are examined for potential risks to Canadians and their environment before entering the Canadian marketplace, and that any appropriate or required control measures have been taken. So the onus on manufacturers and importers is to provide a complete data package for substances subject to the regulations. Required information includes administrative information, substance identity, exposure and use information, as well as a technical package including phys-chem data, ecotoxicity information, and mammalian toxicity data. The onus on the new substances program is to assess the information and take action when warranted within prescribed time frames. And these time frames tend to be quite short, a maximum of 75 calendar days. Next slide, please. So here's a list of the toxicity data requirements. The important thing to note here is that the test data must be generated following GLP. Within our regulations, OECD test guidelines are recommended, although alternative protocols and methods may be accepted. So data generated using NAMs can be used to meet a test data requirement. However, the new substances program can accept or reject the NAM if not suited to measure the systemic toxicity endpoint being evaluated. Next slide, please. Just to further emphasize the issue of GLP, subsection 15.2 of the NSNR states that the laboratory practices to be followed in developing the data must comply with the practices set out in the principles of GLP. And GLP is evaluated by looking for a variety of statements, the date signatures, GLP compliance statements, QA statements, as well as a statement of the study plan, protocol, its deviations, references to SOPs, and we do look for raw data to be included as well. And next slide, please. With respect to alternative approaches such as NAMs, information in support of a new substances notification under the regulations may be obtained from alternative approaches. These alternative approaches will be acceptable when, in the opinion of the new substances program, The approach provides a scientifically valid measure of the endpoint under investigation that is deemed sufficient for the purposes of risk assessment. We do accept data using alternative protocols, and the desired data must be acceptable to the new substances program as well. Sorry, I'm just getting chats popping up here. There we go. The description of the alternative protocol should include a detailed description of the test principles and design, the methodologies and controls used, validation studies of the accuracy and variability of the test method in comparison with the prescribed method, and any references to the protocol in the scientific or technical literature. With respect to new tests, any new test method used to predict human toxicity following chemical exposure must be a good model for human biology and must have a high biological coverage in order to predict systemic toxicity. So we're looking for methods that would predict all phases of ADME, not just metabolic competence. Next slide, please. So with respect to NAMs, as we've been discussing, there are many needs and challenges for regulatory use. Application to quantitative risk assessment can be problematic or is problematic. NAMs offer mechanistic information that's often lacking in traditional toxicity studies. However, making use of that data can be very challenging at times. This is why we'd be looking for standardization of methods, particularly with the interpretation of the data and the link to adverse effects. The development of study guidelines and reporting formats is desired as well. And for new methods, of course, we do need to increase our knowledge, and there is a definite need for training. With respect to validation, given that GLP is mentioned directly in our regulations, we are looking to see if a study method would be amenable to auditing under GLP. Of course, we have concerns about interlab reproducibility of data, and the validation, we're looking for it to use a variety and a high number of test compounds that span a range of physical chemical properties to ensure that they're applicable to the types of substances that we see. Next slide. That concludes my presentation. Thank you very much.
Thank you. Thank you so much, Deborah, for this short but interesting presentation. So we'll move to our next and last speaker for today, Elijah Petersen from the NIST in the US. Elijah, floor is yours.
Thank you. I'm excited to share some about the technical aspect related to validation. And what I'll be describing is based on a manuscript that myself and a number of other colleagues that have their affiliations on my last slide. But this was really a team effort, and based on discussions that we've been having for some of them across a number of years. Next slide. So one of our motivations is really related to how can we fulfill some of these parts of OECD Guidance Document 34? One of the points was related to how do you determine the optimum conditions for each test element? And the second was how can you really understand the limitations of a test method so that could support additional transferability and so forth. The question we have though is how to really go about doing this. And so one of the challenges, or what we have to propose is this technical framework, which I think is designed to be very complementary to the information in GIVIMP and some of the other presentations that you've seen here today. So I'll describe some of the general ideas for this technical confidence framework in the upcoming slides. Next slide, please. So again, as I was saying, this is really a collaborative effort. We had worked with colleagues from CPSC, NIST, DOD, and EMPA. As you've heard many times, we need to have reproducible results. And one of the ways that we went about this is to try to, we were looking at, well, how's this done in other areas? Well, next one. Okay, good. Is to use some of the basic quality tools. And these can really help with adding confidence to measurement processes. So I describe in this talk about how to, in general terms, how to apply these. And it needs to be general because there is such a broad range of NAMs with the different test systems, exposure systems. I mean, it's just hugely variable. So I try to keep things at a very high level. What I won't have a chance to get into today is some of the statistical model building and how to be, to have transparent decisions as a result of NAMs if you understand all the sources of uncertainty. And lastly, there may be trade-offs during the experimental design part where if you, for example, have more controls, you may be able to test fewer test substances during a thing, or it may be that it increases your cost. So there's no perfect answer, but it's important to weigh the trade-offs of different choices as you're making an assay. Next slide. Oh, next one. There we go. So the first step in this technical framework is one that I think Anna really covered on a lot, which is before we even want to begin this framework, there's all kinds of stuff that needs to be done, such as looking at the biological relevance, capacity to fulfill a testing need, and then some assessment of, is there a capacity to have the quality sufficiently improved? If there's some, I don't know, for example, highly unstable reagent in your assay, maybe no matter what we did, what we could do, we couldn't get it up to the right level. But if you have met all these, then we can go through this technical framework, which tries to provide a step-by-step process that people can follow. But if you look at the right-hand side, I have these dotted lines, and that's meant to indicate that this is iterative. It may be that during your statistical data analysis or assessing method transferability, then that you're popped back up and you need to reevaluate some of your conceptual evaluation. So it's not like, I mean, one way you can kind of go through, but then you also may be, they're interrelated steps. It's not like a hard and fast, do once and then you're done with that step. So as you're going through these steps, the conceptual evaluation part, this is all just theoretical. It's based on discussions, reviewing the literature and so forth. You're not really performing any experiments here. But during this step, you can really outline what you think are going to be the key sources of variability and how to have an assay design that you think is going to cover them. But then in the next step, the rubber meets the road and you actually start doing some experiments. It could be that you're, such as doing robustness testing or assessing the applicability domain. And you could be evaluating some of the data that comes out from your assay, such as in the control charts, scatter plots, or histograms. This all then feeds into your statistical data analysis. And if you need it, method transferability. And you would try to design this so for some context of use, you don't need it. For some you do. We're kind of agnostic in this discussion, but if you do it, here's what you could do beforehand to increase the likelihood that you're going to be successful in your ring trial. Next slide. So this is an example of a cause and effect diagram of what you can, and I guess, well, you make it a little bit smaller because I think the bottom is a bit cut off. There we go. Yeah, so a lot of these examples are from the published literature. So you can, if you want to look at the more details, you can dive in. And yeah, I forgot to say at the beginning that the manuscript has already been submitted. We're currently revising it based on the comments we've received. So we're hoping it can come out very soon and we can, read more into all these details. But so for the cause-effect analysis, the idea is that on each of these branches, you have expected main source of variability. Then you have sub-branches, which are some of the particular ways in which this main branch could be important. For example, an assay protocol, in this example with MTS assay, it could be the watching step or your curve fitting and so forth. And what you can do during the robustness testing is to evaluate each of these branches, and ideally the sub-branches too, with robustness testing or your process control measurements, so that you're monitoring the key source of variability. Next slide. Another way to look at at this process is with flowcharts, where you basically just diagram all the steps involved. Now the specifics, and this is from a recently published paper in Toxics. Now the specifics don't really matter that much, but what you should have is that your control measurements should be able to cover all the steps in the chart. It may be that your control measurement may cover a few of the different steps, or it could be that you have multiple control measurements covering a certain step, but the main thing is that you get coverage. So if something's off in one of your steps, hopefully one of your control measurements should raise a flag and you know that something was off there. Next slide. This can then lead to plate design. Again, I mean, there's so many nuances here. I won't get into too much of it, but this is an example from this recently published paper where you can see on the slide, we have a bunch of our process control measurement or control measurements. For example, negative control, positive control, interference wells, and so forth. And as you're monitoring this, you're really going to get a good understanding of what's happening in your assay. And there's trade-offs here. Like, for example, if we had done, I don't know, fewer of the interference wells, maybe could have done other test substance, but we really thought that this was the right trade-off based on the data that we're getting for this assay. Next slide. So this is an example of what some theoretical control charting could look like and things that you could look for. So for part A, obviously this is not looking good. Your mean value is decreasing with time. Also not good in B, where the variability is increasing with time, suggesting maybe there's some change in your reagents or something that's happening that's unexpected. And for C, that's also pretty obvious, where there's an abrupt shift. And you could then look at your check sheets and some of the other things to see, well, did something happen between 5 and 6 that's leading things to be off here. Next slide. This is an example of scatter plots. What you want to have is an example like you have here in part A, where your two control measurements are not related. And on the y-axis is the mean optical density of your negative controls. This is a particular example is from the MTS assay. On your x-axis is then your positive control. There really shouldn't be a relationship between those two, because your positive control is reflecting what you think would happen with your test substances. For part B, there is a clear trend, and so then that suggests that, well, we should figure out what was happening to lower the number of cells for part B, and also maybe have a specification around 1.25 or something, so that you're not having this interaction. Because if you're not controlling, if you're allowing a pretty broad range, you could have the situation for part B where your positive control is varying by a factor of 2 because your number of cells isn't under control well. This is from an example of an interlab study that was published a number of years ago with some international collaborators. Next slide. It's just another brief example of some data. We have kernel density plots. You'll notice that the data is asymmetrically distributed. So that something just happened with my computer. I hope, can you guys still hear me?
Yes, Elijah, we can hear you.
Okay, so could you go, so basically you wanted to see if your data is normally distributed. Could you go to the next slide? Or I'm not able to see the slides at the moment.
We are on the summary, Elijah.
Okay, great. So to summarize, the basic point is that you want to encode the quality into the assay through the protocol. We think that using these basic tools can really help people fulfill some of the motivations I described at the beginning in guidance document 34 and support some of the what's described in GIVIMP, and hopefully overall that this can support the technical validation side of the assays. But again, that should be after it's hit the other points, such as the fit for purpose for different contexts of use and biological relevance. Last slide, please. So I'll mention here my collaborators. This was definitely a team effort and discussions across a number of years. We had Blaza Toman and John Elliott from NIST. We had Emily Ranke when she was at the DOD. She's now transferred to a new position at Innovative. Nicole Kleinstroyer from NIST, John Gordon from the CPSC, and Matthias Rossi, our colleague from Switzerland, who's at EMPA. And that's all I have. Welcome any questions at a later point.
Thank you very much, Elijah. And I would like to thank all of the speakers today for very insightful presentations shared with the whole WNT. And we have a set of questions available in the chat. I've not had time yet to look at them, but we will certainly use them to sort maybe out a little bit the questions that could be asked to the WNT as a follow-up to this webinar and in preparation for a short report on this webinar. You will hear from me in the coming few weeks on the outcome of this webinar. We will certainly discuss within the steering committee this first experience with this first webinar and how we might change or adapt some of the procedures moving forward with the next webinars. As you know, we have the next one on ecotoxicology next week, and this morning I also announced the third one at the end of August. So, we are at the end of the webinar for today, and I wish everyone an excellent day, an excellent week, and talk to you soon again. Thank you. Bye-bye.
Thank you.